Hello, everyone, and welcome to this audio podcast. In this series, or as part of this series, the Chromium Colladax. This is part of the science of, in part of the science of nephrotoxicity. This is the science of nephrotoxicity V3, and this is also part of a special the Crux series. And as in this particular audio podcast, I thought I provide a sort of basis or an overview in relating to sort of research based works that followed on from 2003, in a duration in a duration after or thereafter, in and around 2014. There was works that was updated updated and sort of followed up really in sort of in works that looked at comparing renal biomarkers and their utility to monitor the mainstay of immunosuppression therapy. And the works was sort of research bound or thesis bound based research or works, and was also developed shortly after, in and around 2015 and 16, as a formal sort of book publication, and towards particular book publishers. And so what I thought I'd do in this particular audio podcast is provide a sort of a follow up, a bit more of a follow up in relating to the previous two audio podcasts in this particular series, around the science of nephrotoxicity, but specifically based on looking at a bit more depth in relating to sort of transplant immunobiology and again sort of a bit more sort of in depth in around those immunosuppressive protocols or procedures in organ transplanted, in and around organ transplantation, but also in and around works around a sort of additional overview really around different biomarkers, and also different biomarkers and some additional context in regarding sort of post transplantation or transplant medicine, in and around pediatrics. Pediatrics. From this sort of works, I'm going to highlight those particular focuses more specifically. In doing that, in looking to do that, I will begin with highlighting the some context in and around the sort of in and around once again sort of in and around transplant immunobiology to begin with. So in human beings, in human beings, the major histocompatibility complex, or MHC, is known as known as the HLA, human leukocyte antigen locus, and HLA antigens vary between individuals, making perfect cross matching extremely challenging, or at least retrospectively. Excuse me. And unfortunately, these antigens are also sort of the strongest inducers of T lymphocyte mediated transplant rejection, also referred to as acute mediated rejection, or AMR. And HLA tissue typing has been shown to reduce the frequency of allograft rejection episodes, thus achieving more sort of positive prognoses. And so the knowledge of the interaction, or the interconnectivity of antigens with T cells in sort of the allograft rejection process is generally essential to understanding how immunosuppressive sort of agents work. And so donor HLA molecules are presented through antigen presenting cells to resting T cells that have receptors for specific antigen or antigens. And so this sort of recognition process, by way of the immunological from an immunological basis, activates T cells, which produce and secrete markers, also which are referred to as cytokines. And so, for example, interleukins, and express in this context express cell surface receptors to them. So in context of interleukins, and cell surface receptors specifically, in context of the immunological part of the sort of initial responses, so to speak, can be interleukin, can relate to in regards to interleukin-2 receptors. The lymphocyte colony recognizes donor HLA antigen and differentiates and proliferates under the cell under the influence of interleukin-2, or IL-2, which has been referred to as T cell growth factor. And so cytotoxic T cells, or CD8 T cells, bind directly to donor cells, and look to stick to them, and actually stick to the donor cells, and look to also destroy them at the same time. And so other T cell subsets produce more cytokines. So, for example, interleukin-4 and interferon gamma, which lead to B lymphocyte involvement, B lymphocyte involvement, antibody production, complement fixation, and macrophage infiltration. And the result is a destruction of allograft tissue, which impairs the ability for an allograft to function. And previous exposure of a recipient to other human leukocyte antigens, example as example from, for example, where recipients may have had previous blood transfusions, previous transplants, or where in the case of female recipients, for example, where they may have fallen into pregnancy, or increases the likelihood of allograft rejection. And this can risk, or this the risk can be quantified using what's referred to once again as panel reactive antibody investigations, in which the higher percentage score, the higher percentage score, the greater the recipient's sensitivity to reactive antibodies. And so if the PRA score is greater than 85%, the potential recipient is considered to be highly sensitized and might require additional immunosuppression or immunosuppressive sort of agents and sort of pharmacotherapy. And so, in context of aligned to immunosuppression, in the literature, the choice of immunosuppression currently sort of available, or at least retrospectively have been available in the United Kingdom, is are sort of have been highlighted in sort of transplant immunobiology literature, etc. However, the maintenance regime is tailored to allografted recipients. And so, for example, there are several categories in context of mainstay. There are several categories of the metabolites in context of immunosuppression. So there's calcineurin inhibitors, which are deemed to be referred to as sort of the mainstay immunosuppression pharmacotherapy. Then there's antimetabolites, and then there are sort of corticosteroids, and then there are mammalian target of the what's referred to as the MTOR inhibitor, and in sort of in literature, calcineurin inhibitors, for example, cyclosporin A or CSA, originally also branded as Neural or Sandimmune, and both were developed. The formulations originally were from Novartis and then there's been sort of several derivatives, including Dexamune, Capimune. There's been tacrolimus, and there's been Adaport. There's been Taxany, there's been Vivadex, and there's been another formulation in context of tacrolimus by another sort of organization, another pharmaceutics, as opposed to, for example, one of the original sort of formulations of tacrolimus were developed by Astellas UK, and then another pharmaceutical organization of pharmaceutics called known as Mylan in the United Kingdom, sort of developed another sort of another formulation, so to speak. And so in context of antimetabolites, there is azathioprine, which is a sort of a long-standing antimetabolite that has been used in not just as for example immunosuppression, but also in context of for example, in treatments, in treatment pathways for particular cancers as well. And then there's there is mycophenolate mofetil, which was developed originally by Roche in the United Kingdom, and there's been several other sort of generic brands of mycophenolate, including mycophenolate sodium, and mycophenolate sodium was known as was known to be referred to as Myfortic, and was developed by Novartis in the United Kingdom. In context of corticosteroids, there's one particular steroid where most recipients would probably be sort of prescribed or have, and that is known as prednisolone. And generally, prednisolone is available. It's available sort of generally throughout sort of albeit whether in specialist transplant centers as part of prescriptions that are issued, or are available through sort of pharmacies. Pharmacies, for example, in hospital pharmacies or pharmacies on the high street, according to prescription-based items. And then there's also mammalian, something that's referred to as mammalian target immunosuppression, which are linked to the MTO, or referred to as MTOR inhibitors. And so one of the main ones has been referred to as sirolimus, which is branded as Rapamune, and it was developed was originally developed by Wyeth in the United Kingdom, and it's referred to as MTO, as the MTOR sort of inhibitor, and a sort of a pro and is sort of related to or relating to a protein related sort of MTO inhibitor that acts according to cell acts according to cell growth, as far as immunosuppression is. On this basis, I'm now going to sort of highlight a little bit more around a little bit around some of these sort of some of these particular categories in a bit more depth. And so, for example, interleukin-2 receptor antibodies and monoclonal antibodies. Where I mentioned, for example, where I have mentioned where I have mentioned sort of some of these interleukin-2 receptors antibodies and monoclonal antibodies. Some of these have been mentioned in previous audio podcasts as well, but to place a bit of a little bit more of an emphasis. Interleukin-2 receptor antibodies and monoclonal antibodies. So, for example, basiliximab and dacilizumab are perhaps two of the first interleukin-2 receptor antibodies, IL-2, and monoclonal antibodies, MABs, MAB drugs that were licensed in the UK for the use for the use at the time of recipients undergoing particular forms of organ transplantation, if not generally generally all forms of transplantation. And the monoclonal antibodies are traditionally administered or prescribed before surgery, and they generally exert an sort of an immunosuppressive effect only over the first few particular weeks post transplant, for example, but are licensed for the use of combination in context with a calcineurin inhibitor and corticosteroid, which then form for example the sort of baseline therapy, or can form what is referred to as what can be referred to as triple based therapy or treatments, and but sorry, both are licensed, both are licensed in combination with CSA, or have been licensed with CSA, cyclosporin A. Cyclosporin A is sort of an older generation immunosuppressive that is generally as a form of mainstay therapy was used mainly in between pediatrics and adult nephrology, for example, in context of organ transplantation as well, in the early 70s and 80s and 90s, and then after particular clinical trials that have been developed between cyclosporin and cyclosporin A, and for example tacrolimus or tacrolimus, then through particular randomized controlled trials, tacrolimus was or had been a sort of an immunosuppressive agent that had more sort of potency in some contexts, as well, a sort of better safety profile in context of potency, area under the curve, and also sort of efficacy as well. And so in line with sort of mainstay immunosuppressive therapy, which can be dual therapy or triple base therapy. Generally, monoclonal antibodies work by binding specifically to parts of the interleukin-2 receptor that is only expressed on activated T lymphocytes. Inactivating interleukin-2 helps to prevent proliferation of antigen-stimulated T cells. Now, I'm going to highlight a little bit more around calcineurin inhibitors, and particularly the main two, which is cyclosporin A, older generation sort of immunosuppression, and tacrolimus, and cyclosporin A or CSA and tacrolimus are calcineurin inhibitors that work early in T cell activation. T cell activation involves a series of cascades, and enzyme calcineurin, and the enzyme calcineurin is one of the rate limiting points. And so calcineurin is the target of both CSA and FK506, which is the sort of part of the immune complex, which is linked to the function and the function of tacrolimus, hence why tacrolimus or Prograf as the brand name, for example, or Adaport, for example, are abbreviated by transplant specialists at times to be referred to as FK506. So calcineurin is a target of both CSA and FK506 complexes, which inhibit and prevent transcription of the genes encoding for interleukin-2 and other cytokines that sense the T cell activation. And CSA binds to an immunophilin, which is excuse me, which is a cytoplasmic protein called cyclophilin. Cyclophilin A, and FK506 binds to another class of immunophilins known as FK binding proteins, or FKBP, specifically FKBP12, and CSA5, FK506, and sirolimus, as per mention, as a sort of a MTOR protein based MTOR sort of protein sort of relating metabolite together so CSA, FK506, and sirolimus are metabolized in the liver by cytochrome P450 pathway, specifically cytochrome P450 3A4 iso enzyme, and doses of CSA are used vary according to sort of according to sort of apart from organ specialties, but also in and around where there's variances according to sort of interdisciplinary sort of practices as well. So, however, a typical initial oral dose, at least in and around as documented in literature, has been, for example, 4 milligrams per kilogram twice a day, and doses are adjusted according to pre-dose or trial or whole blood concentrations. And so after 6 months or so, target ranges can be reduced to aim for lower maintenance therapy. And so immunosuppression sort of agents or treatments can be monitored using or measured through a particular laboratory or pathology sort of medicine techniques. So, for example, enzyme multiplied immunoassay, or EMIT, or referred to as EMIT, and typical target or trial levels would be, so between 0 to 6 months, it would be has been documented and evidenced as 150 to 300 micrograms per liter, and over 6 months thereafter anything between 75 to 150 micrograms per liter. So whilst literature has highlighted sort of several sort of effects that can be part of it can be part of immunosuppression. I won't go into too much detail around that. However, most specialist centers, or sort of in context of in context of organ transplantation. Start with an oral dose. Can start with an oral dose of anything of 0.1 milligram per kilogram twice a day for Prograf or Adiport or FK506 for use as primary or mainstay immunosuppression, and doses are adjusted. Excuse me. Doses are adjusted according to trial blood levels, and after a few months, target ranges are reduced to aim for lower dose maintenance therapy generally. And in the original works, there's a detailed sort of overview in context of indications for immunosuppression and laboratory investigations. However, in context of Prograf or Adiport or FK506, typical whole blood trial levels have been documented and evidenced as between 0 to 6 months, anything between 10 to 15 micrograms per liter, and anything after 6 months, anything between 5 to 10 micrograms per liter. And the sort of the EMIT in context of enzyme multiplied immunoassay is generally has been a common method for for qualitative and quantitative determination of immunosuppression, first introduced by by a a company known as SIVA in 1973, and it excuse me, and it was the first homogeneous sort of immunoassay to be widely used commercially. The most widely used applications of EMIT are for therapeutic drug monitoring, and particularly serum as a primary as a primary screen for example abused drugs, or particular metabolites to be determined through or in within urine. And like all immunoassays, EMIT uses antibodies that are specifically designed to bind the molecules of intrigue. So, for example, analytes without binding to other substances in the same sample. So it's a unique feature. EMIT has a unique feature in ability to detect sort of the binding without resorting to a cumbersome separation of the bound component. And this is accomplished by including in a mixture of antibodies and sample an enzyme that is attached to analyte being tested. So antibodies that do not become bound to the sample drug or sample metabolite bind instead to this analyte bound enzyme. So the bound enzyme is designed so that when antibodies bind to its analyte the enzyme is deactivated. Two general examples are given are for example, number one, if there's a high amount of sample analyte present, the sample analyte will bind a large part of the antibodies, leaving also a large portion of the analyte bound enzymes free in solution. And so much of the substrate is converted by high concentration of free enzyme. And if number two is that if a low concentration of sample analyte is present, the small sort of concentration of sample analyte will bind only a small portion of a small amount of the antibodies, leaving a portion of the antibodies to bind to bind analyte bound enzymes and deactivate them. In this case, much of the substrate will not be is not converted, and so essentially EMIT sort of EMIT generally involves, for example, taking a sample of a patient's serum or urine, for example, containing the immunosuppression, with a solution also containing a known concentration of antibody and the enzyme substrate. And then after a short duration, for example, it can be less than a minute, to allow binding, a known concentration of conjugate is also added. And then aligned to that, there's a measurement of concentration and appearance by color or fluorescence or immunofluorescence. And then thereafter, there's a determination a determined point where, as part of the sort of immunoassay, there's a there's a the efficacy or the potency around the immunosuppression can be compared to different concentrations of the metabolite, by comparing efficacy against toxicity, and because EMIT assays are generally quite sensitive, low or borderline results are sometimes difficult to confirm by less sensitive procedures. So, for example, what's another technique known as assay known as thin layer chromatography, and sometimes a gas chromatography, and also high performance liquid chromatography. And some centers or most centers, in context of laboratory medicine, do perform what's referred to as C2 monitoring, which involves measuring blood concentrations 2 hours post particular dosages. And so C2 is thought, or at least retrospectively has been thought to predict the more accurate individual absorption than traditional trial monitoring, and results in a reduced incidence of acute allograft rejection episodes and allograft and reduced rates of allograft dysfunction. Historically, at the same time, sort of the older generation immunosuppression, as referred to as CSA or cyclosporin A, has 50% bound 50% bound to or the drug bound to red cells or erythrocytes, and 10% to leukocytes, and around 30 to 40% to plasma proteins. Only 1 to 6% exists in a free state in context of immunology and physiology, with around 80 to 90% bound to other sort of plasma proteins. And CSA is extensively metabolized again in the liver and bowel under the influence of cytochrome, again under the influence of cytochrome P453A4. So pharmacokinetic interactions with cyclosporin or CSA have been documented to be quite common, and is primarily eliminated by or through biliary excretion, with the median half-life had been as documented between 6 to 8 hours. Again, in this original works, or in the original works, there's an overview in context of the sort of immunosuppression and laboratory testing, investigations around laboratory medicine according to the different forms of immunosuppression and metabolites. And I'm not going to go into detail around that here, but I will highlight that. Um, I will highlight that in part of this, in part of determining, for example, area under the curve, or area under the curve in context of biopharmaceutics and pharmacokinetics. Creatinine as a biomarker has been a long-standing marker for to determine sort of in context of sort of in context of organ transplantation, and in context of where recipients have had organ transplants, and in aligned to the renal, where renal organ has been transplanted. Creatinine and creatinine clearance as a biomarker. Creatinine as a biomarker to determine renal function or renal insufficiency has been a long-standing biomarker, and sort of a main sort of gold standard to measure, for example, glomerular filtration rate. At the same time, before creatinine, there was a sort of a biomarker that was used sort of in some contexts synonymously during the sort of the early durations before creatinine, and it was something that was referred to as inulin. So inulin was considered as the gold standard to measure glomerular filtration rate, and inulin is known to be a freely filtrated through semi-permeable membrane, which has been a strong argument for the absence of binding to protein. And nevertheless, there have been limitations to its use, and because it's relatively high molecular weight, and the molecule is relatively vicious, is relatively relatively viscous, and does not rapidly reach its volume of distribution. And so therefore only methods using urinary clearance with constant infusion rate seem accurate for inulin as a biomarker. So moreover, most of the methods, except particular enzymatic ones, have been prone to interferences with other parameters, and this can be a limiting factor when measuring glomerular filtration rate in organ transplanted recipients who've gone under sort of allografts in context of nephrology. At the same time, Ioxol is kind of was in line with sort of the same sort of time around inulin. Ioxol has been used, and is a non ionic contrast product, or contrast, and was mainly used or have been used for sort of radiological investigations that involve contrast medium to detect pathology around sort of cysts and tumors, and so ioxol's molecular weight is had been molecular weight was known to be quite around 800 or 821 daltons, or Da, and ioxol is chronologically one of the sort of former biomarkers proposed for, or was proposed to be used to determine glomerular filtration rate as well. It can be used in all patients, or at least historically have been documented that could be used, except in patients with or recipients with true allergies to the contrast medium. So its measurement by high performance liquid chromatography was probably one of the most precise compared to other cold methods. So, for example, where there was inulin and thalamate, and ioxol is compared to inulin in some contexts was less expensive as a biomarker, and the cost of HPLC, high performance liquid chromatography, has been known to be quite low. And more important, to underline that an external quality control has existed for IXL measurement, and it can be concluded, or at least it can be to an extent can be concluded, that interlaboratory coefficient for of variation for IXL measurement can also be has also been established as being quite low, for so for example less than 5%. And so due to its low sort of value, this is where, in context of critique, if you like, using serum creatinine or creatinine provides sort of an accurate measure in the assessment of renal insufficiency, and also in the monitoring of glomerular filtration rate according to recipients on particular forms of immunosuppression metabolites. And so the determination of serum creatinine or urine is has been actually carried out by something that's referred to as Jaffe's reaction, where creatinine reacts with something that's referred to as picric acid, an alkaline medium. The alkalinity is provided by sodium hydroxide, or NaOH, and the most important creatinine measurement range for detecting, for example, silent renal disease or renal insufficiency, and in context of immunosuppression, or immunosuppression monitoring, has been between 85 and 150 micromoles per liter, corresponding to estimated glomerular filtration rate of approximately 60 mil per mil. But again, generally that does depend. It does depend on where serum creatinine or creatinine clearance, as per mentioned a little bit in one of the sort of previous audio podcasts in this particular series. Creatinine and serum creatinine in context of determining renal insufficiency can depend on, and generally has or can depend on, for example, age, gender, ethnicity, and also in context of of the kind of sort of calibration that is used as sort of a basis in sort of in and around laboratory and pathology medicine. So aligned to this also, it was Peak and Whiting in 2006 who actually summarized in a sort of an analytical commentary the measurement of serum creatinine and the work of the laboratory the sort of laboratory working group in context of the National Kidney Disease Education Program, which was developed in the United States. And so the NKDEP had published detailed recommendations for improving the measurement of serum creatinine and to assess the performance of current serum creatinine methods. For example, 31 to assess the performance of serum creatinine methods, at least again sort of retrospectively, 31 samples covering a sort of a range of creatinine concentrations plus a number of controls were distributed to six different laboratories across South Australia and Victoria, using a total of 9 different nine different forms of assays and in combinations and each sort of laboratory that was involved agreed to assay the samples exactly as recommended by manufacturers and according to standard operating procedures across two particular days, and calibration materials for each were sort of run as unknowns as part of this particular a part of the as part of a of the of the investigation, to ensure that results reflected the manufacturer's recommended calibration. And so target creatinine concentrations were assigned by assaying ethanol extracts of samples in quadruple using validated isotope dilution mass spectroscopy. Using validated isotope dilution mass spectrometry modality based on tandem liquid chromatography mass spectrometry. And in the original works, there is an overview of sort of the routine creatinine modalities and the sort of the instruments by highlighting sort of the more specifics. However, I won't go into too much detail. I won't go into too much detail around that. And here I do want to highlight some context in relating to sort of the critique around proteinuria, albumin creatinine ratio, cystatin C, and also a biomarker that that had been mentioned briefly in a previous audio podcast, but to emphasize or highlight again the sort of the sort of comparisons of this particular biomarker or these biomarkers against each other, and it was nucleated gelatinase nucleated gelatinase lipocalin, and so it was Shilpak and team in 2011 identified that preoperative serum cystatin C concentrations were documented to be superior to serum creatinine for the prediction of for example where there would be where there would be evidence or indicated injury in sort of perioperative as part of perioperative procedures, and perioperative urinary albumin creatinine ratio is has been predictive of perioperative injury in adults, but not in pediatrics or young people. And so before Van and team in 2009 highlighted some of the issues in using proteinuria as a basis, or at least in some contexts as a biomarker in determining or measuring renal insufficiency. There are sort of clinical issues that had been that retrospectively that needed to be that had to be addressed before unifying global guideline for proteinuria and immunosuppression in organ transplanted recipients, and monitoring and where monitoring can be developed. And lack of data generally has been one of the concerns in context of laboratory medicine. In adults, post excuse me. So in adults, post-operative urinary interleukin 18 interleukin 18, but also interleukin 8 interleukin 18 has been referred to as sort of another a novel biomarker, and in this context, interleukin 18 is that biomarker that's linked to the nucleated gelatinase lipocalin biomarker, and plasma and the plasma around this particular biomarker. The levels peak within have been identified to peak within 6 hours of where recipients are in intensive care units, and within 6 hours of intensive care unit arrival, whereas serum creatinine has not and did not occur. Serum creatinine concentrations did not occur before 24 to 72 hours in comparison. And so Shilpak and team in 2011 also looked to determine urine interleukin 18 and plasma interleukin 18 and plasma concentrations according to the biomarker nucleated gelatinase lipocalin levels, and we're also associated with longer sort of lengths of stay for recipients in intensive care units, for example, and a higher had and there was evidence had highlighted that there was a higher risk of death according to a part of the investigation by this team, Shilpak and team in 2011. But the urine concentration, according to the biomarker used, was not predicted or was not predictive entirely in context of a renal insufficiency or renal injury or associated with adverse clinical outcomes. And sort of by sort of comparison, at least in a sub study, in a pediatrics or a young sub study investigation, urinary nucleated gelatinase lipocalin and urine interleukin 18, but not plasma nucleated gelatinase lipocalin was performed similarly to plasma and urine to plasma nucleated gelatinase lipocalin and urine interleukin 18 in the adult study for the prediction of renal injury and adverse clinical outcomes. In addition, to point out that robust sort of functional measurements of glomerular filtration rate generally still tend to be important for immunosuppression dosing, and an improved platform to detect different sources of biomarkers such as nucleated gelatinase lipocalin in urine has been suggested, and this may be helpful to also monitor sort of pathophysiological changes in context of acute kidney injury development as well. And there was an investigation by the same team, or where they highlighted that Cystatin C generally satisfies many features of the ideal filtration biomarker, and in some contexts sort of outperforms creatinine in detecting minor reduction in glomerular filtration rate. Furthermore, Shilpak and team identify that Cystatin C predicted mortality in sort of patients or recipients apparently with normal, what would be deemed as sort of normal renal function. And so the practical use of Cystatin C as a biomarker of renal function or renal insufficiency and or immunosuppression monitoring in transplanted organ recipients has not, however, been has not been thoroughly investigated in at least where there's been sort of multi-center sort of comparison, multi-center comparison investigations, and so more research around this still are have been required, have been sort of required. And there was an I will put the there will be I will put the citation in around this, but there's more works that follow on from by an author, Abu Kara, or Kakra Abu Kakra, and team, and if novel biomarkers can be proven sort of superior for these particular to meet particular objectives, then they can potentially replace, for example, serum creatinine as a point of as a way to determine and monitor function in and around at least in and around nephrology practices. Currently, however, in those countries or countries where novel biomarkers are available for clinical use, their function to inform assessments on differential diagnosis and prognostic prognoses and associated sort of decisions is probably where initial use where initial use require sort of more investigation, and it is also unlikely that generally between the sort of between scientist specialties, that there is an overall consensus that a single biomarker will be able to provide sort of an adequate monitoring of particular immunosuppressive agents, and a panel specific selection of biomarkers may be required or necessary to monitor titers between for example toxicity, where efficacy meets toxicity, or vice versa. So in closing, I do want to highlight a little bit of an overview in context of immunosuppression in pediatrics. More specifically, so in the case of pediatrics post-transplant and the post-transplant population, dose modifications should be considered where prescribing immunosuppression with known nephrotoxicity, with a known nephrotoxicity profile according to adults. The lower age group subsets of pediatric population may be particularly sensitive to certain excipients that are not entirely inert and may have side effects, and so particularly some excipients which can be used on adults, or can be implemented or used in adults and older, and in sort of adolescents or older children, may be toxic. That may be toxic in neonates because of the sort of immature and rapidly changing metabolic and eliminatory sort of parts of physiology, and the salt the salt of the active ingredient and chemical nature of the preparation, generally according to particular immunosuppression metabolites, must be carefully considered to avoid administration of excessive amounts in and around where there can be sort of a sort of a sort of general sort of a negative interaction, a negative sort of negative negative results or false positive results where electrolytes are sort of part of are part of the process. So in pediatrics, determination of at least historically cyclosporin A by a high performance liquid chromatography has been quite specific. Drug efficacy and monitoring can be separated and quantified. However, this method is longer and requires and has required sort of specialist scientists. And the advantages of enzymologic reactions are their rapidity and automation. Different monoclonal antibodies or MABs also show a low cross reactivity with cyclosporin or CSA metabolites. The fluorescence polarization immunoassay and the EMIT for immunosuppression monitoring in pediatrics in the pediatric population require an initial manual pre-treatment step consisting of precipitation of erythrocytes and proteins by methanol and centrifugation. Then a second immunoenzymologic step is performed in and around the equipment and in and around the sort of laboratory aspects, and then this manual pre-treatment imposes can impose frequent calibrations because of because of sort of weak reagent stability, and liquid immunosuppressive preparations more often contain excipients like preservatives, of which concentrations should be at the minimum level, if not possible to eliminate. And so when preservatives are required, immunosuppression concentration should be at the minimum level, and a thorough justification for the choice of immunosuppression should be is required. Formulation used for particular chronic conditions may cause repeated cumulative exposure to excipients as part of immunosuppression in some regard, and so therefore the acceptable daily intake and safety sort of profiles and limits of excipients for children or pediatrics generally require more examination, particularly for children or pediatrics in and around where renal insufficiency is evident, and so where there's lower renal elimination and where there's evidence of renal malfunction. And so with that, I am going to draw this audio podcast to a close. I am going to highlight this as a side though before I close off, and that is to say where I in a previous audio podcast I did there was some a brief overview in context of what's been referred to as Luminex technology, and so I'm going to highlight a little bit here around that. So Luminex technology technology is now, or has generally advanced. And Luminex technology is built on sort of proven existing technology surrounding flow cytometry, microspheres, lasers, digital signal processing, and sort of traditional biochemistry that have been combined have been combined in a sort of a novel and unique way, and so featuring featuring a flexible open architecture. Generally, Luminex technology can be used to perform a sort of broad range of bioassays quickly and sort of effectively and accurately and generally quite accurately. Luminex. So the bases around Luminex is that Luminex color-coded beads called microspheres are divided into 100 distinct sets. Each bead set can be coated with a different specific can be coated with a reagent specific to a particular bioassay, allowing the capture and detection of specific analytes from a sample. And then within the Luminex compact analyzers, lasers excite generally stimulate the dyes that are used to identify sort of each microsphere particle, and also any sort of reported dye capture during the assay. Many readings are made on each bead set, and further validating the sort of analysis. And so in this way, Luminex technology allows sort of a multiplexing of up to 100 unique assays with a single sample, both rapidly and sort of more precisely. So the Luminex system is or Luminex technology has a flexible analysis based on the principles of traditional laboratory medicine, and even in around academic medicine, where for example techniques have laboratory techniques have included, for example, flow cytometry, that is generally designed to meet the needs of biomarkers of particularly various of particular biomarkers that vary in different sort of in and around biomarkers of different sizes and their sort of concentration as well. And so with that, I draw this to this audio podcast to a close, and thank you for taking the time to listen in on this, and this has been an audio podcast on the science of nephrotoxicity V3. It's been an audio podcast surrounding the special the Crux, part of the Chromium Colladax. It's a chartered edition. This has been the Shientist, and it's been another Storm Robotics production. Thank you for listening in on this one.